Imagine a spiral staircase, elegantly twisting and turning, holding within its steps the secrets of life. This is the double helix of DNA, the blueprint of life for all organisms, from the smallest bacteria to the largest whales. Each time a cell divides, this intricate structure must be faithfully duplicated, ensuring that each daughter cell receives a complete set of genetic instructions. This process, known as DNA replication, is a symphony of molecular machinery working in concert, a testament to the elegance and complexity of life at the molecular level. DNA replication is essential for the growth, development, and reproduction of all living organisms. Without it, cells would be unable to divide and pass on their genetic information, bringing life as we know it to a standstill. Understanding the intricacies of this process is key to unlocking the secrets of life itself. The process of DNA replication, while complex, can be broken down into a series of manageable steps, each orchestrated by a cast of molecular players. From the initial unwinding of the double helix to the final ligation of newly synthesized strands, each step is crucial for the faithful duplication of the genome. Before the double helix can be replicated, it must first be unwound and separated, like the two sides of a zipper. This crucial task is initiated at specific points along the DNA molecule known as origins of replication. These origins are characterized by specific DNA sequences that attract a protein complex known as the origin recognition complex, or ORC. The ORC acts as a landing pad, recruiting other proteins to the site and forming a pre-replication complex. This complex includes helicases, enzymes responsible for unwinding the double helix, and single-strand binding proteins, which prevent the separated strands from rejoining. This initial unwinding creates a replication bubble, a region of single-stranded DNA accessible to the replication machinery. From these origins, replication proceeds bidirectionally, with two replication forks moving in opposite directions, like two tiny bulldozers clearing a path for the construction of new DNA strands. DNA polymerase, the enzyme responsible for synthesizing new DNA strands, has a peculiar quirk. It can only add nucleotides to a pre-existing strand. This poses a problem at the start of replication, where no such strand exists. This is where primers come in. Primers are short sequences of RNA, synthesized by an enzyme called primase. These primers provide the necessary 3' hydroxyl group that DNA polymerase needs to initiate synthesis. Like a painter needing a base coat before adding color, DNA polymerase requires this RNA primer to begin its work. Once the primer is in place, DNA polymerase can begin adding nucleotides, following the base pairing rules, adenine with thymine and guanine with cytosine. The new DNA strand is synthesized in the 5' to 3' direction, meaning that nucleotides are added to the free 3' end of the growing chain. With the primer in place, DNA polymerase takes center stage, meticulously adding nucleotides to the growing DNA strand, using the original strand as a template. The process resembles a microscopic assembly line, with each nucleotide carefully selected and added to the chain, ensuring a faithful copy of the original sequence. DNA polymerase isn't a lone worker, it's part of a larger protein complex known as the replisome. This complex includes other enzymes like helicase, which continues to unwind the double helix ahead of the replication fork, and topoisomerase, which relieves the torsional strain generated by unwinding. As the replication fork progresses, one strand, called the leading strand, is synthesized continuously in the 5' to 3' direction. The other strand, called the lagging strand, is synthesized discontinuously in short fragments known as Okazaki fragments. While primers are essential for initiating DNA synthesis, they are made of RNA, not DNA. These RNA primers must be removed and replaced with DNA to maintain the integrity of the genome. This task is carried out by a different type of DNA polymerase, along with an enzyme called RNase H, which specifically degrades RNA. RNase H removes the RNA primers, leaving gaps in the newly synthesized DNA strand. These gaps are then filled in by DNA polymerase, using the adjacent DNA as a template, ensuring a seamless transition from RNA to DNA. Finally, an enzyme called DNA ligase seals the gaps between the Okazaki fragments, joining them together to form a continuous DNA strand. This process ensures that the newly synthesized DNA is a faithful and complete copy of the original template. As the replication forks progress, they eventually meet, signaling the end of DNA replication. At this point, the newly synthesized DNA molecules separate, each consisting of one old strand and one new strand, a testament to the semi-conservative nature of DNA replication. The termination of DNA replication is not yet fully understood in eukaryotes, but it involves the recognition of specific termination sequences and the disassembly of the replication machinery. The newly replicated DNA molecules are then packaged into chromatin, ready to be distributed to the daughter cells during cell division. Understanding the intricacies of DNA replication is crucial for unraveling the mysteries of life. From the faithful duplication of the genetic code to the development of new therapeutic targets for diseases like cancer, DNA replication lies at the heart of many biological processes.